So, good afternoon, everybody, ladies and gentlemen. Good afternoon, everybody on the YouTube audience. This afternoon, we have a particularly fascinating talk within the Fleck Lecture series. My name is Harald Atmannspacher. I am representing the Ludwik Fleck Center of the Collegium Helveticum at Zurich. And today, our special guest speaker is Professor Paco Calvo from the University of Murcia in Spain. Paco started his career in the late 90s at the University of Glasgow in Scotland. From there, he went to the University of California at San Diego. Hi, Paco.
Hi.
At San Diego. And then moved to the University of Murcia, where he now is the lab head of the so-called Minimal Intelligence Lab, University of Murcia. He's not only doing research on plant cognition or plant intelligence, or should we say plant consciousness. These are all different variations of the same theme, I guess, with different problems and different challenges. But he is also a leading figure in the philosophy of plant behavior and plant signaling. A philosopher concerned with issues at the intersection of plant biology and cognitive science, Paco wants to know two questions, or the answers to two questions, I should say. What, if anything, is there to say about the intelligence of plants? And number two, what is the best way to conceptualize plant intelligence? Now, for us at the Ludwik Fleck Center, it is particularly interesting to know that, or to hear from him, how the challenges of plant science can be analyzed from a philosophy of science point of view. And as we all know, Ludwik Fleck was one of the early figures in a certain movement in the philosophy of science, which is now called relativism. For instance, Thomas Kuhn was one of his big followers. Thomas Kuhn is known by everybody, of course. Paul Feyerabend is a big name of a philosopher who belongs to the same kind of direction, who was here in Zurich for some time. So the title of Paco's talk today is Understanding the Challenges of Plant Science. And the subtitle is Reflections from the Inside Out. And that includes, of course, the philosophical perspective. Paco, thank you so much for being here with us, even though it's only, at the aseptic atmosphere of remote communication. But we are very happy. We're very happy. Yeah, put it away. We're very happy to have you here. And I should say, the screen is yours.
Okay, thank you so much. Let me see if I can share it.
Yeah, we see it.
Yeah. Okay. Yeah, Harald, thank you so much. Hi, everyone there. Hi, Alex in Alicante and everyone else, wherever they are, all over the place. We don't know where they are. Yeah, I guess far too many questions, right? There are far too many questions. You mentioned two, and I could even, lower the bar even more modestly. Maybe not just one, or maybe not even understanding the challenges of plant science, but rather like trying to somehow understand or throw some preliminary light on these challenges, right? We're opening far too many cans of worms, talking about plant cognition, plant intelligence, plant consciousness. So they all come with their own problems, their own toolkits to study them, to analyze them, both empirically and conceptually. So far too many cans of worms, we are just gonna try to open two little cans of those worms, right? And let's see what happens. Well, I guess, it's a shame, actually. It's a real shame that we cannot be there, Alex and I, that are here based in Spain. We couldn't make it to Zurich. But, I'm not going to say that pandemics are a good thing, but at least, being confined, maybe that gives us some time to reflect and, to sit down in our, our attics or whatever. I have an attic like that one in the picture, where I started time-lapsing plants before we set up the lab in the philosophy department. the very first preliminary observations to the naked eye, and both time-lapse and to the naked eye were being done in this attic. And this confinement last year brought me back to those memories of those early days and the excitement of watching plants to the naked eye and all that stuff, right? And one thing is that, okay, maybe we have a good chance, and I mean it, to sit back, slow down, and try to appreciate with a different eye what is it that we are missing. Because certainly there is, I mean, of course, there are evolutionary reasons, especially, flight or fight interactions and many other issues, education. There are all sorts of issues that partially explain why we are plant blind. We don't quite see them, right? So actually, you see this picture, you see the lady, you see the cat, and these are the two guys in the picture, right? I mean, these are the two guys, and maybe you might pay attention to this plant here, but this one definitely belongs to like the lamp or the desktop. It's just part of the furniture, part of the background. But the guys are these two, right? The Homo sapiens and the cat. So there is something we can, and I think something that we should do about it, right? Because after all, they are more similar to us than we can imagine, right? We are closer to each other than it might look at first sight, right? So actually, I think you mentioned reflections from the inside out. I meant to say reflections from the outside in. So the guys in are the plant scientists, right? I regard myself as the philosopher of cognitive science, the guy knocking on their door, say, hey, plant guys, may I join you and see what we can find out together? If you want to know first, before talking about the outside, if you want to know what's happening in the inside, what we find here is that there is this dispute in the plant science community that, well, of course, in history goes back to Darwin and the romantic botany of the 19th century and way earlier, in fact. But for our concerns, we can draw attention in between an orthodox mainstream plant science, so the one that like Thais or people in the mainstream plant physiology literature would represent. Any plant biology student has gone through this book in undergrad school. So this is like sort of like the Bible for plant biology students. And there is something to say about that. We mentioned education. So something to say about all the stuff that we miss by only teaching the mainstream vision, right? On the other side, we have like this group of people. In this case, this is the Society of Plant Neurobiology that got started like 15 years ago. That was a book that came out in those days, Communication in Plants, and look at the subtitle, Neuronal Aspects of Plant Life, right? Then Tony Treguawas came with some papers, again, like 20 years ago, talking about plant intelligence I insist, from within the plant science community, but not from within orthodoxy. So these were like the heterodox guys, the dissenters within the plant sciences. And this is the dispute that have been going on for the last 2 decades. And one of the things that we see is that we haven't learned that much, and I mean it by learning that much in terms of resolving the dispute. So if you go back to one of the first papers on pushing to draw this metaphor, to draw the analogy in between neuronal systems and neuron-like systems like plants, like of course they use, they make good usage of electrochemical information processing pathways, signaling pathways, but of course we don't mean to say that they have neurons, but functionally speaking, the substrate of plants are allowed to play a similar role. So this plant neurobiology view of an integrated view of what we mean by plant signaling and plant behavior was contested, strongly by this orthodoxy, by the mainstream plant sciences. And they say, look, I mean, no brain, no gain. I mean, forget about it. If you're talking about intelligence, if you're talking about consciousness, about cognition, either you have the right type of substrate, in this case, a neural substrate, or end of the story. Forget about it. You are just doing, or we might throw as much light as we can from within the plant physiology itself, or from within the plant molecular and cellular biology, or from within the plant genetics. So of course we can get a lot of understanding of what's going on within plants by looking at those levels of explanation, right, and description. But you cannot go beyond that. So there is a leap, and there's, forget about it, you won't reach it, right? And then that debate, that tension in between the two schools have gone on for the last 2 decades, all the way to the present. And this is again the same group with ties and co-authors explaining that, look, forget about it, plants neither possess nor require consciousness. That might be useful, metaphorically speaking, but I wouldn't like people to focus on terminological, semantic, or metaphorical disputes. So it's not about that, right? We want to discover whether they are actually doing something, functionally speaking, of the same kind that we animals do, right? So it's not a semantic or a terminological issue. We mean to say that consciousness facilitates plant behavior. That's what we respond to these people. And if you look at the, the correspondence in the last few years, the last 3-4 years, this is what I mean by not even knowing if this is a stalemate, or even worse, it might not even be a stalemate. It might just end up being like one group of people, one community, they have their journals, they have their audience, they have their readership, they use their terminology, they do their research, they have their lines of work, and the other guys do their own stuff in their own journals, with their own community, with their own rules of the game. So we have a split in the community, and there is no true communication or true understanding of the challenges for the whole community as such. And that's what worries me, right? Because from a philosophy of science perspective, that's really worrisome, because we might end up simply thinking that we are talking or discussing issues when we are just, into blind alleys, all of us, each one of us on their own blind alley, right? So that's why I put the emphasis here on a farewell to arms. So scholarly speaking, I would say, let's stop these disputes about who said what, and in response, how did you respond, and then in this other response, blah, 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 blah. We end up not understanding what is it about, right? And what is it about is like, if we change these terminological disputes for, they say an image is worth 1,000,000 words. Well, imagine a time lapse made of hundreds of thousands of images, right? It's worth a lot of words. So here we might just check, okay, let's see what these guys do, right? And then we can discuss what's going on. In this case, you are stimulating a plant on the nectar scales, and by stimulating it, you allow the one, now I lost the laser point, but you can read it, the stamens that are hidden underneath the petals, you'll see them on the side here, you can see them here, right? So you allow those immature stamens to move progressively, really slow, of course, that's why you need to time lapse, to appreciate plants at their own time scale, right? But once you do that, you realize that they are way more sophisticated than we thought they might be. Check it out. Right, this is far too slow even for the footage. We can speed it up, and you can see how it's moving in a few minutes. You can actually watch it to the naked eye. Some plants are so fast that you don't even need to time lapse. You can watch to the naked eye in a few minutes how it's progressively making the right sort of movement that is required, right? Of course, this is all very nice. I mean, but plant physiology wouldn't be moved by this, and cocci shouldn't be moved either. We might say, hey, this is just, a very nice example of an adaptation. Of course, no big deal. So we are talking about intelligence. We are talking about the individual. So what happened in its history? What happened to this guy? What sort of experiences it went through that allows us to explain why it's acquired the type of competencies that it has, right? Not what came in their genes and stuff like that. So you might say, well, this doesn't really show much. Then we need to move on to examples where we are actually incorporating or making use of the toolkit of the animal literature. And that's what's critical here. One of the lessons that I was going to say that we learned, but we didn't learn it. But one of the lessons that Darwin tried to teach us all a long time ago is that botany is not subordinate to zoology, right? Botany is not subordinate to zoology. They are on an equal level. And if that is so, we might be able to apply or to borrow the methodologies, the toolkit of the animal literature, the animal sciences, and try to do something similar, right? So this is just an example of what happens when you apply those toolkits. So you can try and find out whether plants can learn. And I don't mean just, for example, habituation. We also know that plants can learn by habituation. Habituation is the simplest form of learning, but here we are talking about something more sophisticated. We are talking about classical conditioning, associative learning, and that's a whole different game, right? Now we are talking business. Now this is not something you can say, hey, remember before the stamen moving into the center of the stage? This is not just an adaptation. We are triggering a conditioned response by pairing two stimuli, right? In this case, by pairing, the blue light, which could be like the food, if this was the equivalent to Pavlov's dog, with a neutral stimulus that would be like a fan here, producing an airflow, and by presenting them in the appropriate manner, you might be able. In this case, it was shown by Monica Galeano, a colleague of us. She's working in Australia. So this is like 4 years ago. She was able to show this, that we could talk of the pea plant in a way as similarly as we would talk of Pavlov's dog. So definitely, if we are able to do that, if that is the case, then this is not simply something we can unearth by using the methodological toolkit of the plant physiology understood from a mainstream perspective. Now I said if that is the case, but is that the case? We don't know yet, right? There are a number of issues. I said we were going to open a few cans of worms. So can of worms, number one, is that what we find in the literature in the last few decades is that at best we have a mixed bag of results. We have certainly negative results where plant scientists try to test for forms of learning, of plant learning, and they didn't quite manage. Then we have some positive results, also in the 70s, and more recently, the paper by Monica that I just told you about. And then we have some early reports in those days that, that there might be some conflations, uncontrolled confounds. It wasn't that clear. So the balance, the scale wouldn't be, pointing in one direction or the other. So, nowadays, what we find, or again going to the very latest, so that you can see how the field has been moving forward, is that the team of this is Casey Markle. He's a postdoc of Lincoln Ties, of the book I mentioned earlier. He has just published this paper explaining that there is not really a very robust evidence about associative learning in pea plants, right? Then you have the response by Monica's team explaining that, that the protocol they were employing wasn't really suited for testing, for condition learning. Then they respond back again. And if this sounds familiar, we are back to the old sterile dispute of responding and responding and responding, and then we say, hey, sit back. Let's think about it from a different angle. Are we doing things correctly? What is going on here, right? Are we trying just to convince ourselves, or who should we convince, right? This takes us back to some of these issues that Harald mentioned initially. Of course, if you go to some issues in the sociology of science and how we collectively, the community as such, is able to make progress. This really reminds us that we should go back to some of this literature and you know, and read it very carefully, because I think some of the tips are already in place once we look from the outside in, right? If we go more recently to, you know, to things like Isabel Stenger's, like Another Science is Possible, well, she has, you know, I love the way she put it in some paper, you know, a plea for slow science. We really, again, going back to the attic and the pandemic, we really need to slow down, and slow down in observing plants, and slow down in the way we present results and what is it that we are doing, right? In fact, for one thing, if you think about it, this is just, if you think of these experiments with pea plants, the animal literature, or the type of methodologies they were, behaviorist methodologies they were employing, they were borrowing the standard Y maze used in rats, right? So you have a Y maze, then you can see whether they are going left or right. So you can do that with plants also. Of course, you have to customize them, because of course plants behave differently. We need to pay attention to the way plants grow and develop. So if we think, so one of the explanations of plant blindness, the fact that we don't appreciate plant life, what they do, is because we think of motion, we think of locomotion, what's fast and furious, right? So, okay, the guy going there, this predator, this prey, so the things that live with us at our own pace. We just need to understand that plant behavior means phenotypic plasticity, and this means growth and development. So we need to be able to pay attention to watch patterns of growth, right? And that's something you cannot do by looking at the Y maze and checking where is it that the pea plant is poking out of, right? So the pea plant is taking a left turn, the pea plant is taking a right turn, but we cannot simply say, okay, did it go left or right? No, we actually need to watch it, right? So you need to see it in motion to understand what is it that it's doing. So this is just an example of a pea plant growing in a Y maze under similar conditions to those reported in Monica Galeano's 2016 paper. So it's not, we are not saying, hey, did it come this way? Did it come this way? And you tick a box. No, it's actually, I need to see how it's actually progressing, right? We need to see what's actually happening as the plant is getting closer to the target, right? So we cannot throw light upon these issues unless we put it in these terms. Let me, okay. Yeah. So if this is this is a truly vexing issue. Are plants able to learn? But think about it. I don't know what the answer is. We are working on it now in our lab. We are trying to replicate Galeano's experiments. We are working hard on that at the moment. We are trying many variations because we don't think there is a clear answer. Either way, the fact that it hasn't been replicated doesn't mean that the phenomenon is not there, of course. And even the fact that it was done in the first place doesn't mean that plants are able to learn by association, of course. So we need more data, we need more replications, and we need to keep working on the topic, right? That's what I meant by slowing down. But put it this way, if just imagine we are able to throw some light, however preliminarily, upon the dispute as to whether plants are able to learn by association or not, right? So if you are able to do that, you have a good argument to move on to discussing plant cognition, plant consciousness. Eva Jablonka, co-workers and many others have pointed out that that such structural anatomical implementations of this type of learning could entail sentience. So think about it, because the emphasis here, if you see what I'm reading, the emphasis here in the case of Jablonka is on the implications of that proposal, so the connection in between associative learning and sentience, for the distribution of consciousness in the animal kingdom, okay? In her case, it's the distribution of consciousness in the animal kingdom. But the question is, what prevents us from expanding and asking about the distribution of consciousness in the tree of life, in the whole tree of life as such, right? So those are two different issues. One is the connection from associative learning to sentience. And a different issue is why that should be linked unidirectionally to one kingdom, to animals, and not to others, right? So, but if the connection is there, we can explore, theoretically speaking, or conceptually speaking, we can explore a number of answers, right? At the same time that we try to make progress on the empirical front. So these two go in parallel, simultaneously. So that's something, that's part of what we are trying to do. I only brought this here, this talk of the construction of apparatus. This is the type of booths we use at the Minimal Intelligence Lab. I don't want to spend much time here, but I do want to raise a concern from a philosophy of science perspective. And it has to do with all these biases, of course, subjective biases, confirmation biases, cognitive biases of all sorts. So we are humans, scientists are human beings, and like the rest, we have our ways to see things and our ways to do things and handle issues. So we don't have a privileged status at all. And we need to be aware of that, because that might explain part of this tension in between these two plant science communities, the mainstream and the heterodox, right? So actually, this is, I just put these pictures this morning just to show you the crew here. This is Adi, he's a postdoc in evolutionary biology. And this is Adrian, he's a pre-doc coming from the States. He works in ecological psychology. And we are putting those two fields together. But here the idea is that if you pay attention to the type of setting, so this is the setting we are customizing to test for plant learning, plant learning. So we time lapse from the side, and then we are able to check how they bend to lateral light, or how they bend to lateral light as opposed to a top light with a neutral distribution, no lateral vector. But the focus here is on the fact, or what I would like to share with them. I tell them every day in the lab that one of the important issues is to realize that we might risk. So this is this is just something else that we'll see that we need to do. We might not be able to throw light upon plant behavior and plant intelligence simply by time lapsing, however informative time lapsing is. So we might need to combine that with electrophysiological recordings. So we also need to check for electrical activity within the plant and see how that correlates, how that combines with the time-lapse information to have a better understanding overall, right? So, but again, this type of gadgets that we need to make to DIY in the lab to throw light upon behavior drives me to think about the IKEA effect. And that's true. I mean, if you have to assemble this furniture that you bought in IKEA, you very probably will fall in love with what you've done, right? So we need to be aware of that type of issues. Otherwise, we will want to show that plants learn because we've spent so much time building these toys that we need to really show it, right? So step back, slow down, chill out, reflect, and that's part of at least the philosophical reflections from the outside in that we need to make sure are understood within the plant science community, right? But getting into the core of the issue, going back to this connection in between associative learning and sentience, animal or not animal. When we talk about sentience, this comprehends, of course, different types of experiences. We can talk about stereoception, we can talk about interoception, we can talk about proprioception, and if that is the case in the animal domain, in the animal kingdom, the very same thing happens when we talk about plants. We can talk about interoception, proprioception, stereoception. This is just an example of gravity sensing. We well know that plants, if you tilt them over, they will bend and they will grow again, the roots following the gravity vector, positive geotropism, shoots growing upwards, negative geotropism. So we do know that. And we have a better understanding of how that works. It's just a matter of sedimentation, right, with the statocytes. So as you shake the roots, those sediments relocate, they resediment at the other side, and the signaling response allows for the type of phenotypic plasticity that we see when they do actually change course of action as they grow. So you can relate that to the pattern of growth in roots, in shoots, whatever, right? This is an example of roots growing upwards and then suddenly turning downwards, right? And this is interesting. This is from František Baluška's lab. This is interesting because we might say, hey, plant behavior, is not that relevant or doesn't take us to plant intelligence because it's not flexible. It's just hardwired. And it's not hardwired. It's only hardwired because we don't look at it the right way. So with the right type of experimental designs, we might be able to appreciate how flexible it is, right? So in this case, when we put roots upside down and we put them to grow in these thin tubes and they have to grow upwards, they grow upwards. And then as they are growing upwards, you may, hold on. When they are growing upwards, like here, you may say, well, no big deal. They are simply growing upwards because they don't have a space to make a U-turn, right? There is no enough room for the roots to make a U-turn and follow the gravity vector as we know they should because of the statocytes sedimenting, right? But the fact is that responses are not one-to-one, are much more complex and flexible than we thought. So they have to integrate all these informational channels that don't have to do just with gravity, but with light and many, many, many other things, as we'll see now. So here, since roots are photophobic, as soon as you put a top light, being photophobic, now they will find that there was enough room to make a U-turn and escape from light, right? So responses are not that hardwired, are not that inflexible, and the connection that we make is that plant behavior, apart from being adaptive, is flexible, is anticipatory, and is goal-directed. So if plant behavior is anticipatory, is flexible, and is goal-directed, then we are talking about plant cognition and intelligence proper, or we have a good reason to put up or to make the case that is what's happening, right? So plants, we know, are sensitive to all sorts of signals. In fact, this is just a little list we put up recently just to have them all together. But if you go through it, you will see that, there is all sorts of channels dealing with energies impinging on the plant's surface from all over the place, from root to shoot. And the plant critically needs to provide a global response, not a response on a one-to-one basis to gravity, to light, to whatever, right? So it's much more complicated than that. So if we think about if we put it in these terms, the problem that we have is that from a plant physiology perspective, all we do, or we usually do, is to compare human or animal physiology, different systems in the animal physiology, to different systems in plant physiology, and simply put the emphasis in the fact that they are more broadly distributed. So of course we know that plants, since they are rooted, one of their evolutionary strategies is to divide and conquer. So they don't have a central system, everything is distributed throughout the whole plant because they cannot flee. Being rooted, okay, if you chop one branch, I will grow another branch, right? So they have different ways of dealing with the contingencies that are impinging on them. But the mistake is to think that this is all that is happening here. So it's just not a matter of being more or less distributed. It's a matter of understanding the vascular system of the plant. If we look here, so we are here putting the emphasis on the vascular system. So when we are talking about the distribution of resources, the allocation of resources, the water and nutrients and minerals and sugars being pumped up and down throughout the plant body, throughout the flowing and xylem pathways, so that vascular system is not just understood physiologically speaking as the way to send sugars, minerals, and water throughout the plant body, but as an information processing system. So that is the key, to move to the plants, to a cognitive science of plants, to plant cognitive science. So it is information processing what we are after, right? So once you put it in these terms, we can talk about, you know, how complex information processing is in plants and how complex the substrate is. So this is just a picture of the vascular system, the vascular flowing pathways of a papaya stem. This comes actually from a book from J.C. Bose in 1926, a book entitled The Nervous Mechanism of Plants that was completely ignored, of course. And a century later, we still have to say, hey, we need to go back and rediscover all this literature from the late 19th century and the beginning of the 20th century to understand that they do have the type of structures that would allow them to implement those types of competencies at that level of sophistication as required once we understand that their behavior is truly flexible, is truly anticipatory and goal-directed, right? So now, if we do that, this is important to understand from a cognitive science perspective. I would like to say that there is no one methodology that should be followed against the others. So you can move from, you know, now I'm putting here just a few of them. If you think of a classical, you know, reductionist or representationalist or computationalist approach to the human mind, well, you might follow the same steps when you talk about plants. I'm not saying you will reach very far. I'm just saying that methodologically speaking, you can follow the same rationale because plants are not that different. If they are processing information, we should ask, are they computing? Are they representing, right? The very same way we do with animals. We can treat them as a parallel distributed processor. So you can apply, you know, the toolkit of neural network theory. You can put the emphasis on embodiment, on distributed systems, on the interactions in between the vascular system. So just remove here the nervous system and talk about the vascular system, right? So the vascular system of the plant with the rest of the plant body and with the rest of the environment, both socially, that means with other plants, but also with pollinators, with all the guys out there, right? We can talk about 4D cognition. So we can talk about plants in a framework of cognition being extended, embodied, embedded, and active. So the very same paradigms that we are familiar with from the cognitive sciences, I'm saying, let's not discard any of them. Let's use them all and see how much light we can throw upon plants from them all, right? This is just an example. Here we did something from a predictive processing approach, which is representational, right? It's a representational approach. But what I'm saying is that we don't need to sign the blank check that we are going to follow one and only one methodology. So in fact, what I'm going to show you is that we are going to the other extreme in this case, to ecological approaches that are non-representational and non-computational. And by being non-representational and non-computational, they might be shedding or they might be throwing a distinctive light upon plant behavior and our understanding of plant behavior. So you can go from a framework, a more classical framework understood in, you know, constructivist terms, like, you know, talking about indirect perception, so percepts in the head of the model of the organism. You can talk about the cognitive doings, the algorithmic level of description, or you can go for a more ecological approach in which you put the emphasis on the direct perception, the direct interaction in between the organism and the environment. You put the emphasis on the behavioral and not on the cognitive. You put the emphasis on the issues of implementation. So the particularities, the idiosyncrasies of the implementation, the vascular system of plants, and how much light that can give us as to the inner doings and how we explain plant behavior, right? So we can explore all those methodologies from the more representational to the more radically non-representational and non-computational, right? This is what we do when we follow that. So if you think in terms of ecological psychology, if you think in terms of an approach which is direct, which is not inferential, not constructivist, which is implementational, which is all these labels we were using, then this is the setting we use at the Mintlab, right? So we place a climbing beam. I said one of the issues is goal-directed behavior. So we want to know whether the plant is actually approaching a target in a goal-oriented manner, in a controlled manner. So not simply that the plant keeps growing and eventually manages to twine around the pole because it bumped onto it. Oh, I keep growing and I bumped onto the pole and I happen to grab it. No, it's that we want to understand or we want to find out whether the plant is controlling the approaching maneuver, controlling how it grows towards the target, right? So to do that, you can see one example here. So just pay attention now to the picture on the left. So forget about this picture on the right. Just look here to the zenithal, to the top camera. Now you can see here a plant performing anti-clockwise pattern of circumnutation, right? So it's moving around the clock 360 degrees, and it's growing and growing and growing and bending and bending and bending. And somehow, up to a point, a working hypothesis is that there is some transition from aimless growth to goal-oriented growth, right? So suddenly this pattern of circular circumnutation will change. Now it goes to the back, and you will see how it fishes straight to the front, right? So that transition to fishing, that pattern of growth is providing us a nice hint. I'm very cautious here. I'm not saying I believe my eyes. I say we have a good case to keep working, to keep trying to understand what's happening here, right? See what I mean? We don't want to buy that without being self-critical, because we don't want to fall in all those biases I was mentioning, all those subjective and cognitive biases, right? But think about it. Think what might be happening here. If you look at this petal, from our perspective, that's what it looks like, right? Well, it might look rather differently when you put it under ultraviolet light. And surprise, surprise, this type of ultraviolet light is what you need if you are a pollinator. This is what the petals look like from a pollinator's point of view that they can see in this wavelength. So from their perspective, this is pretty handy. This is pretty handy because it's providing the platform, the type of information that is guiding the approaching maneuver. So the insect, the pollinator, has these cues. There is the generation of an optic flow that has to do with the couple system. So the pollinator flower system as such. So as the pollinator moves with respect to the target, these energy flows are providing, in this case, is visual information, is an optic flow, but is providing information as to how to land. So here the plant is making it easy for the pollinator to land, okay? So think about it. Now we are putting the emphasis, as usual, from our anthropocentric and animal-centric perspective, we are putting the emphasis on the animal. Oh, is the animal that the guy that is, is helping himself to the ultraviolet landing marks to land gently on the petal? No, let's switch roles. So who is pulling the strings? Let's switch the roles. This is a pattern of coevolution. So the role of the plant is as important as the role of the pollinator. So this idea of exploiting ecological information, like the optic flow generated as one system moves with respect to the other, is something that you can exploit equally from the point of view of the plant, right? So the plant itself, think about it. This is just a classical example of how you can rely on visual cues to explain the control of the standing in humans, right? So this is a classical papers from the 70s by Dave Lee, one of the fathers of the idea. And let me show you, oops, let me show you here. The information, this is the swinging room, right? So the guy here is controlling, with this handle, is controlling the walls. You see the walls are not on the floor, they are hanging from the ceiling. So it can control how the walls move back and forth. So by move, you see the walls move backwards, and that's making the child fall, right? So it's affecting the child the way so it's affecting because it's changing the size of the walls, the image in the retina, right? So it's those relative changes that do matter, right? So think that if we switch roles, if we put the stress on the plant as a subject and not as an object, then we can play a similar game, and we can say, hey, aren't plants relying on ecological information the very same way that animals do, the very same way that the pollinator does? So we might say, well, this is a clear example when you think of shade avoidance. Plants sense, so you know, red light is being absorbed, far red light passes through the plant, so they can rely on the ratio of red to far red light. So that relative value, the ratio of red to far red, is ecologically informative in relation to the pattern of growth. So you can be growing under the canopy, and you can see how that ratio goes up or down depending on which direction you grow. So if I'm growing in this direction and the ratio is going down, I know I'm gonna find neighbors in the neighborhood. So I better change gear and grow in another direction if I want to find sun flecks or a window to do photosynthesis. So you see, now is the plant that is playing the role of the pollinator in this game of exploiting energy flows and ecological information, right? We are not talking about representations in the head of the plant. We are not talking about computing anything. We are talking about the direct perception of possibilities of interaction with their surroundings. So if we talk about those possibilities of interaction, if we talk about the affordances, what the plant perceives, then we can understand in the same way with this methodology what was happening in the case of the climbing bean and the pole. The climbing bean wasn't perceiving the pole as a static object that is sitting there. It's perceiving climbability. So an affordance, an ecological affordance, is perceiving the possibility to climb it, and is generating information by circumnating around. So the same way that when I grow one way or another, the ratio goes up or down, and that's informative. When I grow around, when I circumnate like a climbing bean, I'm generating energy flows that are going to be informative for me to control the pattern of growth to make it goal-oriented towards the target. You see what I mean? So this is the way to go about it. This is one way to go about it, right? So because we didn't want to commit ourselves to just one single paradigm. But from an ecological psychology perspective, we can do an ecological psychology for plants, right? We are not doing plant physiology, we are doing psychology. It can be cognitive psychology or ecological psychology, but it's plant psychology, right? Now let's move on. So that was the first can of worms. I don't know how good I'm doing in terms of time, Harald, but 10 minutes? 10 minutes? Okay, good. So let's move on to the second can of worms I wanted to open. Consciousness, sentience. For some reason, a few years ago, I was becoming increasingly concerned about the fact that we might be missing it all despite being right there. It could be right there and we don't see it because we haven't trained our eye. And I thought, hey, could it be possible that we are treating them like locked-in syndrome patients? I mean it. Couldn't be there, trying to flag us. Hey, I'm here, I'm alive, I have mental life, with all the square quotes that you want to put to that mental life. Remember, you can apply many different frameworks to understand mental life. But hey, I'm here. I'm just locked-in syndrome patient. right? So if you think of in terms of the awareness of the environment and the self and the different levels of arousal, and you think of all this in continuum of alertness from a coma to these cases, well, it might be, you know, we might have a nice connection here to you know, to dealing or trying to throw some light. Now I'm talking experimentally by using anesthesia. I mean, this is what happened. I mean, when I took this Venus flytrap to the veterinary hospital in Murcia, the University of Murcia, they thought I was nuts. I said, hey, do you have some isoflurane? Can you help me out and put my Venus flytrap to sleep? And they were like, what? How do you mean? Yeah. I mean, the same anesthetics that you use, the vet uses to put cats and dogs and horses to sleep in the vet faculty. So this is their place. I took my Venus flytrap there. We put it under a glass bell like this, and you just stick some cotton pad with anesthetics, and wait for a while, and you see that the response flattens. So it does flatten. I mean, they just become unresponsive. So here we have something very interesting. Something is happening here really interesting. Just think about a Venus flytrap. If you trigger the inner hairs, of the Venus flytrap, you will see that by triggering it twice, you've got to trigger it twice within a certain interval of time, like 20 seconds. But if you trigger it twice within those 20 seconds, they do that the twice within 20 seconds to avoid false positives, something accidentally touching it. But if you do it that way, they close. But what happens? What happens when you touch it? After having put the plant to sleep. If you put the plant to sleep, now this is an anesthetized plant. The plant doesn't respond anymore. You can touch it and touch it and touch it, and the plant won't close, right? You might say, hey, you messed with the plant so badly that it just, it fried out. It won't recover again. But no, the plant does recover. So the plant does recover. So now you can trigger it again when the plant has come out of anesthesia, right? Now it closes again. If only you will see that it closes slowly, slightly slower. So you might say, hey, it came out of anesthesia, it's still kind of, hey, what happened? And now it's responding again, but not as fast as it was responding at the very beginning before you apply the anesthetics, right? You can try that on different plants, right? So this is Mimosa pudica before and after anesthesia. Now it's not responding. You can do it with the Venus flytrap, and you can do it with pea plants. So this is pea plants down here, non-anesthetized, anesthetized. So the very same molecule that you use with animals puts to sleep live across the tree of life, right? So that, again, from a plant physiology perspective, that might be something like, oh, wait, so what? We have the physiological explanation of why that's happening, right? So we have the physiological explanation of why that's taking place. You're messing up with the membranes, you are not allowing for the action potential, and it's not being triggered. So plants aren't firing spikes of voltage anymore. At the electrophysiological level, we do have a good explanation, but they might stick from mainstream plant physiology to that reductionist, mechanicist explanation without trying or without wanting to see further, right? But again, we can do the very same thing. In fact, well, this is something we have, it's not out yet, we submitted it. We are working on this idea, because actually, I mean, when you can do the same type of metabolic maps that you can rely on the same tools to map this metabolic activity throughout the plant body. You can do fMRIs, you can do a plant PET scan. So you can use the very same tools with exquisite spatio-temporal accuracy to see how water is traveling throughout the whole plant body, what's happening in the vascular system, at an amazing level of precision, both spatially and temporally, right? So you can do that, and then you can explain many things about rates of photosynthesis, about stomata closing and opening. You can relate that to environmental contingencies. There is a risk of dehydration. Plant leaves might be closing their stomas. So all these things might be understood, and we could have a good metabolic map of all those inner doings of the plant. And yet, we might be missing what's interesting about it, which is the Coxside perspective applied to plants. So when you do that, this is an example. We've put forward an idea of what could be happening from an integrated information approach, right? So this is not just, we are sort of advancing the type of test, experimental, empirical test that you can do. So we could actually try and measure level of sentience in plants, right? We can do this from this perspective. We could do it from some other perspective. Again, it's not a privileged status. We have different theories of consciousness. We don't need to rely on this one in particular. We could rely on many others, but this is just an example for the sake of illustration. So if you can work on the application of magnetic fields, we know plants are sensitive, of course, to those magnetic fields. So you can induce some perturbation into the plant system and see how that relates to the level of arousal, to their sentience, right? So that really connects to anesthesia in a very interesting way. We know we can put them to sleep. We know they come out of anesthesia, and we can say, hey, think about it. If subjective experience in the plant, if they have it, if it arises from interactions between the parts of the plant body, then we might be able to probe plant sentience by perturbing the plant with magnetic fields and try to mess up with the vascular system, with the root to shoot vasculature. So these vascular pathways I was telling you about that related to all the physiological activity, from an information processing perspective, they could be really valuable in letting us know what's happening when we perturb, magnetically speaking, when we perturb the system, and see what remains integrated and what remains decoupled under anesthesia and without anesthesia. So we can have a way to measure those things, and we are not making anything new. We are simply putting on the table the very idea of going back to Darwin, not subordinating botany to zoology. If we can do that with animals, there is not any reason that prevents us from doing it with plants. That is the message, right? Regardless of the outcome, regardless of the results, right? So I'm finishing now. Yeah, perfect timing. But I couldn't help bringing something from Sidney Brenner in his autobiography, My Life in Science. He says something very interesting, and I found it delicious. I always use it in my philosophy of science undergrad course. And I tell my students, look, knowing too much curtails creativity. And knowing too much, by knowing too much, I mean knowing too much from within one paradigm, not allowing other questions to be raised. I'm not talking about setting in stone the answers they provide from those other frameworks. I'm just talking about the very raising of the questions themselves, regardless of the answers, right? So that's my reflection from the outside in, not to curtail creativity. And thank you for your time.
For your time. Thank you. Thank you so much, Paco, for this fascinating and interesting talk. Now, the setup that we have prepared for you is we have a panel here with 1, 2, 3, 4, 5 people. And before we go into the Q&A session, I just want to introduce to you the members of the panel. It's okay, Paco? Yep. So to my left, that's probably to your right, if I'm correct.
To my right.
This is Meredith Schumann. She's from the University of Zurich, and she's working in the Department of Ecology and Geography. She's very interested in she has a special focus on plant ecology.
Oh, that's great.
That is great.
I'm actually in chemistry and geography.
And in chemistry. Oh, I'm sorry.
With a focus on plant ecology. It's okay. The
focus was right.
Yes.
Good. And she's the head of the remote sensing lab.
I'm not the head. I'm in the remote.
Part of the. So I did everything wrong.
Okay.
Hopefully next. Okay, yeah. The person to my other side. This is Alexander Borbely, who is well known for his decade-long work on sleep research, but he's also an expert in psychopharmacology, also University of Zurich. Then on the far right is Jürgen Kornmeier from the University of Freiburg. He is his specialties are cognitive neuroscience, in particular bistable perception paradigms, learning, and also neuropsychology. And last but not least, we have our invisible visitor. That's Alex Gomez-Marin. Alex, are you here?
Yes.
Can you hear us?
Yes.
You can hear
Sure.
us. He didn't manage to activate his video, but he will be here orally.
He's together with me, the other guy missing the beers and dinner.
Okay. Alex is at the University at Alicante in Spain, and he works on the behavior of organisms and the neurobiology of organisms. So this is the brief outline.
Great.
Now, who wants to start with questions or comments?
I have a very general one virtually.
Alex,
here.
please, go ahead.
Okay, let me start with a radical and perhaps weird question to Paco, especially since he's a scientist, but also a philosopher and a philosopher of science. So roughly 400 years ago and before, one would claim that people, and that would include intellectuals, whatever that means, saw the cosmos as essentially alive. Basically everything, plants, rivers, galaxies, planets. Now, today some people would say, well, they were just stupid or naive or simply pre-scientific. But isn't it curious to you, Paco, the amount of effort we need now to argue for sentience, intelligence in ants and in plants? In other ways, what's the null hypothesis that plants are not? Isn't that really strange? And who really has the burden of proof?
Yeah. Of course, I mean, when I think about it, in my heart of hearts, I said, who could deny it? I mean, to me, it's so obvious. I mean, they couldn't be sentient. They couldn't not be sentient, right? Simply because you connect from day one sentience to life itself. So I'm not talking about, I mean, you have theories of consciousness, the cellular basis theory that relates it to, prokaryotes, unicellulars. So anyone will have sentience insofar as there is life. So that might be obvious to many people, but you know what? To me, the problem really has to do with the top-down approach from the Homo sapiens, the Homo sapiens stance. So that's something we have been fighting against for millennia, of course, and this is one of the examples where I said we are simply cross-talking each other. So, and here is an example where terminological disputes, if we solve those disputes, might help, because to some people, the use of words they are very sensitive to the use of certain words. So when we talk about the nervous system of plants or the phyto-nervous system of plants, they go crazy. Say, plants don't have neurons, anathema. So they say, take it easy. If you don't like the label, swap it, change it, drop it. Just change the words if you don't like them. Let's talk about the tests. So let's test it. Let's talk about the predictions. Let's talk about the models that allow us to throw that empirical experimental light. And but the problem is that they cannot, that's my impression. Many times we, all of us, we cannot dissociate our hunches from the linguistic labels we use to make our case. And that's maybe one of the reasons why we can't help being defensive of those labels, intelligence, cognition, consciousness, and say, hey, plants, why don't you use another label, okay?
Yeah, Meredith?
I have a list that I would... Yes.
Microphone, yeah.
Yep. I'll try to make it short, and I would be really interested in which points you would like to focus on.
Okay.
And I think it also relates quite well to this last question. So one thing, so I come from a background of chemical ecology, and that involves working with people from many different disciplines, and we have to be quite careful about how we talk about our experiments so that we are doing the same experiment, for example, when we are working together, because it's very easy for that not to be the case. And in most of my work, I am measuring phenomena that happens at several different layers, one could say, and it's very important to know why I am measuring these different layers and not to conflate things, right? So a tool that I learned early on that's been extremely helpful has been Tinbergen's 4 questions. Do you know these?
No.
So this comes from behavioral ecology, actually. It comes from the early tradition of behavioral ecologists, and the idea is that in biology one can ask how and one can ask why.
Right.
And
okay.
Under each of these, right, there are the proximate and the ultimate causes, yeah?
Oh, sure.
Right, okay, so you're familiar with this, but this is come up again also as levels of analysis. For example, there was a short article by Paul Sherman in Animal Behavior in 1988, right? And the Sherman article in 1988 was pointing out many examples in which I think the same kind of talking past each other happened in the animal behavior community. He used very provocative examples in order to get a response from people, but his point was there are these people posing hypotheses about physiology.
Hold on a sec. I need to close the window because it's getting... Oh my God. We are getting a hurricane here. Okay. Okay. Good.
Right. So there are people posing hypotheses at the physiological level, at the level of physiological mechanisms, and there are others posing hypotheses at the level of function. And these coexist. One cannot be used to falsify another. These are different levels of analysis, right? And I don't understand actually why plants should be special. So any organism is functional and active. And I also think that any organism that has a circadian clock makes predictions and adjusts those predictions based on experience in a very flexible manner, and that there are known genetic and chemical mechanisms to do that that can provide evidence. So these are the general thoughts. I wanted to end on 2 points. One is there's also a lovely article. I think it is called "How Would a Molecular Biologist Fix a Radio?"
Right.
That you might be interested in if you don't know it already. And that's also a bit the point, that it might be that, I don't know if I'll get the examples right, but the main point is that the molecular biologist is never going to figure out that the plug of the radio is unplugged. No, but we'll spend a lot of time naming different parts of the radio for things that they don't do. Right? So this is also a bit about a holistic view versus a reductionist view, which is, I think, what I'm hearing from you with ecological psychology. And then finally, also going back to the circadian clock and going back to levels of analysis, I was missing chemistry in your experiments, and I wonder if you are interested, because I think that it's also far from undisputed, but it's clear that plants flexibly emit and use a huge variety of chemical signals, and it's also possible to tell plants very specific things using specific chemical signals, and this is clearly involved in between plant interactions and also cross-kingdom. Other cross-kingdom interactions, not just humans with plants, but lots of other animals with plants, right? So I think that's I don't know if you're talking to them in a language that they care about, you know?
I understand you perfectly. Yeah, good point. Yeah, you made you've made a lot of points, right?
I told you I had a list, and then I have one very last thing, which is a question, which is I just don't understand what unlimited associative learning is, and depending on how it's defined, I am not intelligent, because I certainly am limited in my ability to associatively learn things. So I would be interested to better understand this operational definition.
Cool. Yeah, good. Thank you. Are you done?
Huh?
Are you done?
Yes, I'm done for now.
Okay, fold your shopping list and put it in your pocket. So thank you so much. Plenty of interesting questions. So let me, most of what you said at the beginning, you used different terminology coming from different disciplines, but they are, after all, we might be talking, because of our different backgrounds, we might be talking different languages here, but I think it all boils down, in a sense, to two issues. One, well, of course, one is the relation in between disciplines. I mean, in this more holistic or more reductionist understanding in between disciplines, of course, there my take is very simple. I mean, we truly need to go for a holistic emergentist approach. We cannot reduce, we cannot reduce the psychology to the biology, to the chemistry, to the physics. So we cannot have that cartoon version of the unified view of science picture, right? But that's not, I think that's not what's interesting here. The other side is what, I mean, once you buy that, or once we all buy that, we are still stuck in this never-ending dispute of function versus structure, algorithm versus implementation. These two levels of description, the how, the why, the where, all these questions you were raising. Of course, the classical, if you go to the Coxai orthodoxy, the classical example is Mars, 3 levels, right, of description. But even in the terminology you were using, and it doesn't really matter, what we are talking is about a trade-off in between function and structure. And of course, they've got to inform each other. So a structure has got to be informed by function, and function has got to be informed by structure. Or if you want to put it differently, you cannot simply work algorithmically without paying attention to details of implementation, and you cannot simply care about details of implementation without bearing in mind the algorithm you're after, right? Now, with that being said, it makes a huge difference whether your commitment to a computational framework is of one flavor or another. So if you are so to speak, a good old-fashioned artificial intelligence person, or you believe in computations and representations under the computer metaphor in the classical way. So if we think that a system can only be intelligent insofar as it's following a recipe by following an explicit set of rules, a recipe book to do things with symbols, if it's about rules and symbols, then we have a problem. But not plants, we have a problem with any form of life whatsoever. So that's what I was telling before in the case of Alex. If we go back to if we raise the bar that high and get obsessed with symbols and with explicit rules, then there is no game. We cannot get the ball rolling, right? Now here there is a finer grained discrepancy, which is whether, having granted that we need to see how function and structure inform each other, we might end up realizing from, for example, an ecological psychology framework, when we work within ecological psychology, these ideas of direct perception where we don't need to posit any percepts, we don't need to talk about information being stored anywhere within the individual. We simply talk about how those energy flows resonate within the individual, and we care about one and only unit of analysis, which is the organism-environment as such. Organism, environment, all together. That's the one and only system we are analyzing. So any level of description needs to honor that level, right? The level provided by the system in itself, right? So once you do that, you might end up realizing that it becomes meaningless to talk about the algorithm. So we need to separate the way function informs a structure we need to separate that issue from an understanding of an algorithmic understanding of that higher level of description, right? So if you are able to do that, you are separating your agenda, your research agenda, from the research agenda of, I would say, a huge part of the cognitive sciences, which are very much obsessed with the algorithm, regardless of how much attention they still play to the implementation, right? So here is where you have some room of maneuver to make a stronger focus, to put more emphasis on those connections without or getting rid of all that talk of computations and representations. Now, if certainly, there is room, we couldn't turn a blind eye to chemistry, that's for sure. In fact, we pay attention to it real close, because as you will know, we couldn't talk about plants interacting with their surroundings and ignore volatiles. So, VOX, volatile organic compounds, we have molecules, airborne, traveling all over the place and allowing plants, to perform kin recognition. The amount of things they do is truly amazing. And the more we know about the stuff they do and how flexible it is truly astonishing. Now, by looking at the chemistry in itself, we might be missing those connections to the coxide approach. So you must say, okay, you work, you know what this molecule does, end of the story. No, we need to think, for example, about, there is one beautiful example. Some plants, when you have this caterpillar, this caterpillar munching on the leaf, they segregate these chemicals and they mess up the neurochemistry of the worm, and they become cannibals. So the worm, the caterpillar ends up eating another caterpillar. They eat in between them and leave the plant alone. Some other plants, use those chemicals to attract a wasp, which is the natural predator of the caterpillar. So they will say, hey, come here, wasp, eat the caterpillar. Oh, thank you so much. So they are using chemically those bodyguards. So it's a chemical, so of course it's chemical warfare or defense mechanisms all over the place. Now, what's interesting here, this is a great example, because you can see how it's not about sticking to the chemistry in itself, but about using a working hypothesis against other working hypotheses on the same footage. And this is an example. Some plants can mimic, I could show you, but it's not really worth it. But they can mimic real nice the phenotype of the host tree they are climbing onto. So this is a parasitic plant, and it's actually Bochyla trifoliolata is called. And Bochyla, this plant, is twining around, is climbing onto a host tree, and for the sake of camouflage, it's mimicking itself, and its leaves take the form, the shape, the type of color, the greenness, exactly the same. They look exactly alike the leaves of the host tree. Now, one working hypothesis is that they do it chemically, that is VOX related. Okay, these molecules being emitted allow all these mechanisms to be, this genetic expression to allow for this phenotypic plasticity to take place, right? Some other working hypothesis has to do with horizontal gene transfer. So of course, you cannot ignore the chemistry, you cannot ignore the genetics, but other working hypotheses have to do with, for example, some form of protovision, that plants might be exploiting some form of proto-image that is not accurate as in the type of images that we exploit with our camera-like type eyes, mammals or vertebrates, but is the type of image that, however poor it is good enough for the plant's own purposes. And here, when you compare one hypothesis against the others, now we are not talking about the level of chemistry as the discipline that runs the show. We are talking about different working hypotheses that we need to tell in between them, following the scientific method, without preconceptions, without prejudices, and one would point to information that relates to light sensing proteins, another would relate to information that relates to volatiles, airborne volatiles, another would relate to horizontal gene transfer, whatever. But the working hypothesis and the model, and this is what is important and many people are missing in the plant science community, the model that we should care about, or that is allowing us to put these predictions on the table and test for those hypotheses, is a model that can only come from the cognitive sciences. Because if it came from plant chemistry, plant molecular biology, you would miss the very question we are trying to raise. You wouldn't ask, how is the climbing being controlling, in case it is, controlling the approaching maneuver to the target? So those questions, you need this Coxeye framework to raise them in the first place. Then we will resort to chemistry, biology, and many different levels of description. Yeah. So,
now, what about limitless flexibility?
Yeah, sorry. So her last point on unlimited associative learning, right? Yeah. So I think, well, I didn't use Eva Jablonka's example. I don't need to agree with her on that. I was just trying to show the connection from some form of learning to sentience. Then we can discuss across the tree of life how it bears, right? But how limited or unlimited that competency is, if you can generalize across the board or not, reminds me a lot of the debate, classical debate in cognitive sciences about, the capacity of a system to behave productively, systematically, compositionally. So all the language of thought discussions, the dispute in the 80s in between the language of thought and neural network theory, right? Where you say, hey, can you generalize in this unlimited sense of behaving adaptively, come what may, once you've acquired the competency, once you follow the rule, can you apply the rule in any context whatsoever? And I don't think we need to buy that. I would be happy with a very limited form of associative learning, which still provides me an entry point to the very idea of sentience, however limited that idea of sentience is, right? So,
thank you. Thanks.
Let me bring up again the associative learning study, because I've been a co-author of Monica Galliano's paper, very distant one. And as you pointed out, there is still some controversy about this study. And I'm very glad that you said that it needs confirmation and repetition. And if I understood you correctly, you are working along those lines. So I think that's very important because one finding is not enough. It needs to be replicated. But let me make another point. As a sleep researcher, I'm struck by not only by the fact that plants have many things in common with animals and humans, but that we have also many things in common, or some things in common with plants. Namely, when we sleep, we are in a sort of a vegetative, plant-like state. We don't move any longer. We don't eat, so we don't take energy. So we are, in a way, resembling temporarily plants. It's also the vegetative nervous system which is then taking over. Proprioception is still there, but exteroception and interoception are no longer present. So it's a very peculiar state where there is some resemblance with what may happen in plants. And this brings me to something which Meredith has already mentioned, the circadian rhythm. We have that present in all animals, even in single-cellular organisms, and of course also in plants. But you mentioned that interesting anesthesia experiment where you are trying to somehow put plants to sleep, although anesthesia is not properly sleep. Did you think of using the circadian rhythmicity as a more physiological approach to put plants perhaps in a less receptive state, because plants also change in receptivity in many functions. And so it would be very interesting to try to, instead of anesthesia, to just exploit a circadian change for your purposes.
Yeah, thank you so much. That's a great question, and yeah, definitely. We are working on that actually, and that's so interesting. So well, first on Monica Galeano's and co-author paper with you, and definitely. I'm a very good friend of Monica. We exchange information on a regular basis. And I'm well aware of the potential pitfalls. I'm well aware of what's happening in this dispute. And we are doing our best to throw light from the outside. So that's definitely very much needed. One thing that worries me is that sometimes people say, revolutionary things, right, really radical ideas like associative learning in plants require revolutionary or extremely solid or robust evidence. Sure, but that's the ABC of doing science. We shouldn't be that concerned about it. In fact, we might end up realizing that sometimes when you find a hurdle, that hurdle might allow you to see somewhere else, to push your own limits and say, hey, maybe what they were doing, there is still an alternative explanation to account for that Y maze directional response of the pea plant. But that's precisely what's allowing you to envision a different design that precisely points out to a solid, robust enough evidence backing up associative learning. Right. So we don't know. This is an open empirical question. And my fight, because I talked to them all. So I talked to Monica, but I also talked to Casey Markle and to the others that failed to replicate it. And I talked to them all to try to make them understand that this is not a fight in between scientific communities, in between ways of understanding plights. This is a fight against ignorance. So this is all of us together against or trying to understand the phenomenon. So I don't need to show or to prove that plants can do X, Y, or Z. I only need to be open-minded enough to explore it, whatever the outcome is, right? But certainly that's a line of research we are definitely, I have this postdoc in the lab pushing in that line, and we are definitely looking forward to having results to share. Now on definitely on sleeping pattern and circadian clocks and stuff like that, that's absolutely wonderful. Yeah, we have in fact here in Murcia, these things. This is amazing because once you are used to. Okay, I need to talk to this guy from plant biology. I need to talk to an expert in circadian rhythms. And then somehow you don't realize that it's closer than you think it is. And I only found out last year that one of the leading experts in phytomelatonin is just next door. Is a guy here in the plant biology building on campus. So I knocked on his door. And last year I said, hey, I couldn't believe you didn't know about my work. I didn't know about your work. Let's go for coffee. And we did that. And it turns out he's being able to, plants synthesize phytomelatonin. So when you take melatonin to go to sleep at night, when you take a tablet, a supplement to go to sleep, plants do that. Plants synthesize their own phytomelatonin. And what's interesting, remember I said that we are combining time-lapse observations, time-lapse footage with electrophysiological recordings. Now here, this is interesting because legumes, like plants from the legume family, like climbing beans, the ones one of our plant models, they perform nictinastic movements. So that their leaves fold at night. No, so this is the leaves. So at night they go like this. They fold, right? So this leaf folding behavior is, of course, it's completely tuned to planetary cycles, day-night cycles. And it's so interesting to look at the circadian clocks throughout the tree of life. So it's no surprise that life on Earth is ticking to planetary rhythms. So we are all dependent on them because they are so informative. And temperature drops, and humidity drops, and the lights go off, by night, sleep tight, sweet dreams. We all do that, right? And plants, it's interesting because precisely that's a very good point. We need to, so we call it phytoacupuncture because we use acupuncture needles as electrodes. So we insert those acupuncture needles, the electrodes, into the stem of the climbing bean, make the electrophysiological recordings. We collaborate with a Brazilian team who are the experts in this. So they analyze that data. But what's interesting is that we can apply phytomelatonin to the leaves and it's being absorbed. So that's definitely something we are doing at the moment. Well, we have the project, we need to get started, but the theoretical framework, it's in place, and definitely looking at patterns of disruption in information processing related to leaf folding that relates in turn to day-night cycles might be very informative in a more ecological setting, much more ecological than applying anesthetics to plants, which we don't know what else is being messed up with, right? So definitely that's
thank you.
That's a great problem.
Thank you.
Paco, we're approaching the end of the session, but before we close it, really, I want to give the word over to Jürgen, finally.
So, okay. Thank you, Paco. Very fascinating talk. I want to make my argument in 4 quick points. So, and my central statement is, of course, plants don't have brains and plants don't move, clearly. If you look in the evolution of invertebrates to vertebrates, you see a continuous accumulation of neurons, and they accumulate at this part of the body, which is the front end, and this is due to the motion of the organism. So this was my first point. My second point is, if you go on the animal part, the very simplest, one of the simplest systems is a combination of a sensory neuron with a motor neuron. So you get information and you immediately react. We talk about a reflex. Some people, including Holk Kruse, which was a former biology professor in Bielefeld, they think that the invention of the interneuron, which means a neuron which is neither sensory nor motor, is the key step for cognitive evolution, because then you can disrupt the reflex. You know, so you can sense information, but you can treat it. And nowadays, the complex organisms, they have more interneurons than sensory neurons or motor neurons. So there is a lot of thinking about the sensory information, or as Freud said, Probeplanung. So testing some motor concepts before actual doing it, you know, so thinking about it. So this is somehow the invention of the necessity of deciding, because you can try out several things and then you have to decide which route you choose, right? So, and Holk Kruse said in one of his articles, or he cited one of his colleagues, and I have to reread it, maybe thinking is like imagination of motion. So my point is now that maybe cognition, as most of the scientists understand it, is closely coupled, closely tied to a moving organism. And maybe that all features we are collecting in the moving animals, which we attribute as cognitive features, and we may look now for animals sharing those features, maybe this is the wrong approach. I wouldn't say that animals are not cognitive, but maybe it's a question of terminology. And let me end my statement with a very nice story. When I was in the train coming to Zurich, I read the New Yorker article, which you may know much better than me. And there was a story from Stefano Manusco about how he came into plant cognition, and he told the guy from the New Yorker that he was seeing Star Trek. He was quite a fan of Star Trek, and then there was this alien which had a much faster time constant. So this alien processed everything much faster than we do. And then this alien entered the Earth, and he looked at humans, and he noticed that they are static. So he treated them, he looked at them like we look at plants.
Definitely.
And maybe what you are talking about is that the critical point is maybe the question of terminology, and maybe the factor time is very much important. So this is my point.
No, thank you so much. Yeah, definitely. So you said a few things. Let me get started with the very end. I would say that intelligence or cognition is about movement. Or I can buy that. I can buy that because plants do move. Their movement takes the form of growth and development. So they don't locomote from point A to point B, but they reach point B. Right? Okay. They stretch themselves until they reach their target. So what's important here is put it this way. What's important about movement is not the locomotion itself, but the type of information that you generate, or not that you generate, that is being generated. So think of the infant in the video that we saw, that it was the walls that were moving. The infant was static. It was the walls that were moving.
Okay. Mm-hmm.
So in that case, being still, there was a relative rate of change in the retina images as the wall was approaching and the image was expanding on the retina, and then that information is what's ecological, what's meaningful for the infant. Think of gamers of video games. I mean, a gamer that is playing video games and is stuck to the chair, like my son right now, and then it's gaming. Then they bump onto a wall, and they have the feeling of the bumping because the pixels on the screen are getting bigger, and then it's on me right into. So we always need to think about it relationally. So it's always relational properties, and if it's relational properties, then there is not any difference, functionally speaking, in between locomotion and growth, because in growth happens exactly the same. Those energy flows are changing constantly round the clock, not because of the agent doing something, but because of the agent environment level.
Can I add something? But this may be motion, but at least it seems not to have made it necessary to accumulate something which we call neurons on the animal side.
Oh, so right? let's go to that part of your question. So okay, so now you mentioned the hub, the neural hub. When we talk, this is something I didn't mention. We have this other thing hasn't been published yet, but here we are sort of guessing. This is just a hypothesis. We are guessing that if we make those perturbations that we were talking about towards the end of the talk on integrated information and see what happens, we have the hunch that the meristematic tissues at the shoot tip and the root apex, so at both ends of the plant, are where information integration is taking place.
I see.
Why? And that's the functional equivalent to what you were saying. if you look at the meristematic tissues, where you get all the new cells, and they get specialized, and then you get all the traveling downwards, the plant body, right? So if there is an area go to the root apex. Just behind the root apex, there is an area that František Valuška coined the expression transition zone to call it. The transition zone is an area of 100 and something cells in between the meristematic tissue and the elongation zone. So where the cells elongate and rigidize. So if you think of the root is like, imagine a root is growing like this. If the root turns left or turns right, that's because of differential elongation, right? So these ones here are elongating more, and then it turns this way. So that, of course, we know the differential distribution of auxin, of phytohormones, and we have an explanation of growth hormone. But what's interesting from an information processing perspective is that those cells within the transition zone are the ones where you find the highest level of zinc firing. So all those guys fire in zinc. And that's, informationally speaking, very meaningful, because as those cells are being pushed by the meristematic tissue, as new cells are being created, those cells keep traveling downwards in the root from the transition zone into elongation zone. As they cross the border in between transition and elongation, they elongate, they rigidize, and then boom. So imagine as the cells are crossing the border, if they all crossed at the very same time, they would elongate at the same time, so they would grow in a straight line. So to have a change of direction, so oriented growth, you need one cell to cross the border at a different time so that you can have differential elongation. So that's being controlled by this hub where we find this highest rate of zinc firing. So it does make sense when you were talking about this bridge in between sensory and motor neurons. It does make sense that at the meristematic tissues, where you get the sensory caps, the sensory apex, both at the shoot, at the tendrils, and at the roots, is where the plant is being more sensitive to vectors of information from the environment. And all that information is being conveyed and it's being exploited by letting it go into a population of cells, a population of plant cells that electrochemically is playing a role which is analogous, in the sense that the outcome that comes in terms of growth is what allows the plant to generate those energy flows. Remember the plant growing and the ratio of red to far red light dropping. Oh, I will turn the other way. So this turning this way or turning this other way is mediated by those cells next to the meristematic tissue. That would be, to me, the functionally equivalent. With that being said, this is just a last comment, Harald. With that being said, I would add that we should be very careful, or we shouldn't be too committed to always drawing analogies, because sometimes they are useful, and sometimes we might say, hey, why don't you just think about plants and forget about animals? Right? So maybe sometimes analogies help, and some other times they might be not necessary, or leaving you somewhere else, right? But anyway, yeah.
Coming back to timescales.
Yeah.
Coming back to timescales. You know, there's one special thing about Swiss clocks. They never go faster and they never go slower than just right. And I think we are just right at 6 o'clock. Thank you so much, Paco. Yes. Thank you so much for your presentation and also for the lively discussion. I think that was very interesting for everybody. And maybe there is something to follow up on it.
Yeah, thank you. Well, thank you to all of you. It was great.
Thank you. Thanks.
Yeah, have fun.
And bye-bye. Bye-bye, Alex, to you too.
Thank you so much.
Bye-bye.
Thank you.
Take care.
Thanks, bye-bye.
Bye.